Hello and good evening to everybody. We're approaching 100 people signing in, so that's a good time to start. Welcome to the third of this series of 5 updates on where we are with COVID. Tonight's topic is vaccines, very appropriate on the evening that the government announced 10 million people had received the vaccine. The format will be the same as ever. So we have a speaker who will speak for 15 to 20 minutes, and then we'll have a question and answer session again for 15 to 20 minutes. Please keep your microphones on mute and your videos off. That improves the connectivity. Place your questions in the Q&A. If you type them in, then they get relayed to me, and then I can pose them to our speaker. And just to remind you that the whole session will be recorded and will appear on the RCPath website sometime next week, so you can always catch up if you've missed the first 2 or if you want to revisit this one. So with that background, it's a pleasure to welcome Professor Peter Openshaw to give tonight's talk. Peter is a professor of experimental medicine at Imperial College. He's also a consultant in respiratory medicine. He has spent time on SAGE, and he's also spent time on the Joint Committee for Vaccination and Immunization, and he's currently a member of NERVTAG, which is the New and Emerging Respiratory Virus Threats Advisory Group. So absolutely the right person to be telling us about vaccines. He's also been president of the British Society for Immunology. So with that, I'll hand over to you, Peter, and we look forward to your talk.
So thank you very much for the invitation. I was asking Professor Irving about the audience, and you're clearly pretty expert, so I won't go into a lot of background detail. I think everyone has become quite an expert on COVID these days, but before I get into some slides about the vaccines, I just wanted to point out that this is by no means a passive virus in immunological terms. You know, this is a virus that has got intimately acquainted with the immune system, presumably in bats, but is well capable of regulating the host immune response to its advantage. And it does that mainly by interfering with the innate recognition that is activated as soon as the virus gets within cells, and it does this at several different stages through these non-structural proteins. And so you can see these non-structural proteins here. So it's, in terms of, why is that important? Well, in terms of vaccination, of course, the vaccines don't do this. They don't interfere with the host immune response. So, there is a prospect of actually making vaccines that cause better immune responses than the actual virus itself. So in terms of what sort of immunity the virus itself triggers, well, it triggers a B cell response and a T cell response. And in terms of, what sort of responses, there's been so many publications, we couldn't possibly go into them here, but I wanted to just emphasize this recent paper, which came out in Nature, where they were looking at the durability of the B cell memory response. My pointer isn't very big. Let me just see if I can make it better. Hang on. Yeah. So the B cell memory response does seem to be pretty long-lasting. It recognizes, mainly the spike protein, but also the nuclear protein. And I think the particularly interesting thing to me about this particular publication was that they did intestinal biopsies 4 months after COVID, and they found in 7 out of the 14 volunteers that they could actually find evidence of presence of the virus. So, maybe this virus is actually persisting and causing some persistent stimulation of the immune response. Okay, so let's now go straight into talking about a few of the vaccines. So there's many different vaccine tracker websites. This is one I quite like, New York Times. where they're looking at the vaccines as they come through the pipeline. This is just the ones in clinical testing, phase 1, phase 2, phase 3, those with limited or early approval and full approval in the US, because this is a US site, and those that have been abandoned. So maybe a point to pick up here is that, you know, not every approach is going to work. And I think it's such a remarkable achievement that we've now got 10 with limited or full approval in this single, you know, for this single agent within a year of its arrival within the human population. Just an amazing accomplishment. And, you know, you can't assume success because some approaches have actually not worked. So just amazing in terms of vaccinology. So what sort of approaches are there? Well, I'm sure many of you will be very familiar with this, but the exciting one for us vaccine immunologists is the ability of injected RNA to trigger an immune response. The RNA gets into the muscle cells and other cells within the muscle and translates protein, which is then recognized, and maybe the RNA acts as a sort of auto-adjuvant, you know, stimulating the TLR system so that enhances the immune response. I remember when DNA vaccination was first described, you know, watching with incredulity that the injection of DNA would actually induce an immune response. But, you know, we had no idea at that stage that RNA was going to turn out to be such a useful vaccine modality. You know, that was in the days when we were used to vaccines being grown in various ways, you know, eggs or tissue culture, whatever, whole virus being inactivated, injected, or maybe proteins, subunit purified proteins being injected with adjuvant, more conventional approaches. And those have been relatively slow to deliver this time compared to the recombinants, mostly the adenovirus recombinants, whether they're chimpanzee or human adenos, some of which are quite sophisticated in terms of being replication deficient, for example, with the Oxford vaccine. So all of these approaches being attempted, but so far not intranasally. And as a mucosal immunologist, I would really love to see intranasal vaccines. But these are given intramuscularly, induce a systemic response, which can be profiled and then tested in phase 3. So just to remind you, I'm sure you're all familiar though. So the Moderna and the Beyondtech Pfizer RNA vaccines, the Oxford AstraZeneca, the Sputnik V, which are coming through, and I think the Novavax results are pretty impressive too. So lots of different vaccines, different choices. Just to illustrate some of the data on which all of this is based. So seems to be almost every day now that we get new key publications coming out. So this is the Chadox, the Oxford AstraZeneca vaccine. And you can see here the ELISA response to the prime and the prime boost. And by comparison here, we've got some convalescent sera. So the sort of levels of antibody which are being produced, measured in this assay, are sort of in the range of the types of antibody that you see following natural infection. So it doesn't exceed natural infection, it sort of matches it. The dose that they gave in this trial is relatively low, even when they got the dose correctly adjusted. They weren't going for the highest doses. They didn't go as high as, say, the Sputnik V, which is about, I think, twice that dose, where they were maybe less concerned about some sort of low-level side effects. This is one of the RNA vaccines. This is the Moderna vaccine. And here you've got the control recovery sera from convalescent COVID cases. And you can see that now we've got levels being induced at the 3 different dosages in this phase 1 trial, which really are at the top end of the range of antibody that you get after natural infection, and which are very tightly grouped. So there are very few individuals who don't develop these strong responses. In terms of the side effects, you can see that in the second vaccination, the side effect profile is rather worse than in the first, and we've also been noticing this in the Beyondtech vaccine. So this is some efficacy data with the Chadox vaccine, and you can see that the control here, which is this is still labeled wrong. In the proofs that were uploaded, they got the labeling wrong between which had the meningitis control and which had the Chadox. And I'm sorry I haven't changed the slide, but basically this is the recipients of the active vaccine, and you can see very good suppression of disease. And in the control group, the infection rate just goes up and up. So there is an accumulation of cases, even in those who have received the vaccine, but it's a remarkably effective vaccine. If we look at this Beyondtech vaccine, so this is the RNA vaccine, you can see that after about 12 to 14 days, you can see an expanded scale up here. Essentially, you get no more cases. So very, very effective at preventing infection in those that received the actual vaccine. So next, this is something which has just come out, which is looking at the reactogenicity of vaccination given to people who are already seropositive. And this is a preprint from Florian Kramer and his group, and collaborating with external people. And you can see that people who are seropositive here, you can see they're seropositive even before they get the vaccine. So they develop a very good, very strong, and well-maintained response just to a single dose of vaccine, which exceeds that after a single dose in those who are seronegative. But you can also see down here that the people who were seropositive because they'd recovered from COVID and then they were given a dose of the RNA vaccine, that they actually get quite bad side effects. If you compare particularly these systemic side effects here in the seronegatives and here in the seropositives, they get really quite bad side effects. And I think this accords with what we've been seeing in the UK in people who've had COVID and then they've been vaccinated. You tend to get rather bad side effects. So we could go on, but I thought I'd then move on to just talking a bit about the vaccine task force and what was chosen by way of vaccines. So the task force basically set about commissioning a lot of vaccines, not knowing which of these were going to be successful, and ended up with 345 million doses. So, enough vaccine, if it all works, to cover the population many times over, even if we have 2 doses. And, so far we've got really quite a lot of vaccine, which is available both from the Oxford AstraZeneca, the Beyondtech Pfizer, the Moderna, and now it looks like we're going to be having some Novavax as well. And we did commission 60 million doses, which may take a little while to come through, but we've got an awful lot of vaccine. So a bit more about how these compare. So this is an interesting graphic from the BBC showing how much money was invested in these different vaccines. And it surprised me how much money had gone into the AZ vaccine, considering that this has been done on a not-for-profit basis. And AstraZeneca have agreed to produce this vaccine at cost for the duration of the pandemic. But that's an awful lot of money that's been put into that when you compare it with the amount of money that's been put in with the close competitors. Now, this is the big one in terms of investment. So thank goodness that paid off. The Pfizer-BioNTech some government money, but a lot of that private money as well. The Moderna, mainly government money, but if we look at the prices, so the Moderna RNA vaccine is coming in as one of the most expensive, so around $30 or so. If we look at the Pfizer Beyondtech, that's about $20, and then the Oxford AZ is about the price of a coffee in a fancy restaurant, I guess. But the price variation is very considerable, and it seems that the companies have negotiated rather different prices at different locations, which may be why Israel has got such a wonderful vaccine record, is that they're paying over the odds for the shots that they're getting. So this is the JCVI vaccination groups going from 1 down to 9. And here we've put on the cumulative population. So, and this vaccination is to prevent 1 death. This is from, I think, the Society of Actuaries. They've worked out how many people you have to vaccinate to prevent 1 death. And you can see that if you get down to about this level, the sort of 4th group, you've got a very good ratio between the number of doses given and the number of lives saved. If you get down to just the general population, you have to give 47,000 shots in order to prevent a death. That doesn't mean to say it's not worthwhile, but when you look at it in those quite hard terms, I think this illustrates just how much the JCVI focused on trying to reduce deaths as much as possible. So it does look like we're going to be able to get down here pretty fast towards vaccinating all of those extremely vulnerable, and then we should see a reduction in deaths and also, I guess, in hospital admissions. Although I would say that a lot of people who are in intensive care are not necessarily in those identified high-risk groups. Okay, so this is a graph of the daily number of COVID vaccine doses administered per 100 people. We're doing exceptionally well. I've actually missed off Israel and Bahrain because they do even better, but they have a very small population, obviously. But I think that our vaccine task force really has to be thanked in part for putting us in the position of being able to give so much vaccine. And also, I guess, the infrastructure which has been built up in this country in terms of vaccinology. So, I think we are doing very well, and news comes out all the time. One of the controversial areas that you'll all be aware of is the decision of JCVI to delay the second booster dose until up to 3 months. And this has just come out showing that the vaccine efficacy actually is, if anything, rather better if you do build in that delay. So I think that this publication, once it comes out in full, actually will give good support to that decision from JCVI, although it was much criticized. And I think from the immunology point of view, waiting to give the booster until the plasma cells have disappeared out of the peripheral circulation and gone back into their memory niches and bone marrow is very rational. And I'm sure many of you, if you've tried to raise anti-sera at any time, will know that if you try and boost too fast, sometimes you can actually get a much less good effect. Okay, so what about problems that we can see arising? Well, this is a slide just to illustrate the many different mutations which are being picked up in the variants of coronavirus. So with it first moving into human population and out of bats, you can, I suppose it's perfectly easy to anticipate that there would be some adjustment of the genome as it settles into human circulation. And certainly, mutations in the receptor binding domain will potentially threaten not only binding of antibody, but also possibly binding to the receptor. So those could be disadvantageous as well as advantageous to the virus. But then there's other domains, 4 domains in total on the spike protein, all of which can potentially mutate and which could contribute to escape from immune pressure. So I guess at the moment, the B1.1.7 variant, which arose in southern UK, in North Kent, and then spread widely and is now in about 4 different countries. That particular variant probably was driven mainly by transmissibility, although there is variable evidence that it may be slightly more severe in its presentation. But that's not really confirmed as yet, but does seem to be more transmissible, but doesn't appear to be able to escape from antibody pressure, either from natural infection or from vaccines. But it's very successful. I mean, this is an example of a graphic showing just how this variant has actually replaced previous strains. So maybe if we have a lot of this B1.1.7 virus around, the Brazilian, South African strains just won't be able to outcompete, not until we've got high levels of community antibody positivity, I guess. This is illustrating the increase in admissions in different hospitals. So COVID patients were climbing in Medway, and then in Dartford and Gravesend, and then various other hospitals. And that really coincided with the spread of these new variants. So here's your South African variant. So this one really took off in South Africa and has become a very dominant strain. And we're still really gathering much more information about this in terms of its severity and whether it can escape well from immunity. It seems to be partially resistant to the antibody generated from previous infection, but that and the Brazilian strain are a big worry. So just last few comments before we open up for questions, but I think this is an interesting one. You might think that in the UK there's an awful lot of vaccine people who are doubtful about having vaccines, but this is the share of people who would get a COVID-19 vaccine if it was offered to them. And we are pretty good here. And I hope that a lot of the public engagement, which has been done by members of the immunological community and clinicians and members of SAGE and NERVTAG, will have contributed to the high level of public information about vaccines and to this relatively positive attitude. But our close neighbours over in France, there's an awful lot of people who are very doubtful about having vaccines. And to me, this is potentially one of the major things that we have to contend with. It's not just the science of vaccinology, making great vaccines is all very well, but if people are doubtful about receiving those vaccines, then that can lead to failure of vaccine programs. I thought I'd just finally highlight this one publication which came out recently, which was actually from somebody who was a philosopher, who was talking about motivated ignorance, rationality, and democratic politics. And essentially saying that, well, a number of interesting things, including that that having a strong irrational belief can actually have a positive benefit to a person because it gives them ownership of, or it gives them admission to a particular social group who share that consensus belief. And that if you drop your extreme view and start proclaiming something more rational, you may actually have a very high personal cost in terms of them being excluded from the social group that you are part of. And trying to work with that sort of understanding seems to me vital if we're going to be able to get vaccines out into the communities where it's really needed and often where there's very high rates of COVID. So what are the prospects? Well, so natural infection does prime for T and B cell immunity, but the virus itself is quite good at immune modulation, and there is hope that vaccines can do better than that, and some of them do appear to. I think it's great that we've got such a rich landscape of potential vaccines, but there's still quite a lot of uncertainty about whether we're going to have to repeat these vaccines every year, whether we're going to have to reformulate the vaccines to match the current circulating viral variants, which does seem, I'm afraid, very likely. Whether we're going to be able to make vaccines which are effective against a number of different diseases and maybe have a mixture, a cocktail of RNA, which might protect against influenza and coronavirus and who knows what else. I think we're all aware of these stories of immune augmentation, and we're just concerned about that. But so far, I would say that there's not too much evidence that that's going to be a problem, but it's something that is in the back of our minds. So thank you all very much indeed.
Thank you very much, Peter, for giving such a concise overview all the way from the molecular biology through the cost to the actual problems of getting the vaccine into the patient. There's been a whole plethora of questions, much more so than in previous weeks. So I think you've actually answered one or two of them. So I might just pick up on those questions that you haven't touched on. I don't know if you could know the answer to this, but we have somebody in the audience who has long COVID and is asking, is there any evidence that if he was to be vaccinated, that would improve his long COVID, or I suppose even possibly make it worse?
Yes, a fascinating question. And I've been asked this many times. And I've been trying to think, if it was me, would I run the risk of having the vaccine and potentially finding that my disease was augmented? I think probably I would. But you would be running the risk that what you're suffering is some sort of immunologically driven disease that you could augment. I think we need to see more data on this, and I would love to know if people. I have heard anecdotes of people who've been suffering from long COVID who have been vaccinated, had a bit of a reaction, and then they've got better. But how common that is, I don't know. Maybe it's something that we need to start accumulating clinical anecdotes and then mount a trial.
Yeah. Okay. The vaccines that we're all using now are all S vaccines only. Are there any coming through that are more representative of the whole virus, and wouldn't that be in the long term a better way to go?
Well, so the Chinese are producing vaccines which are whole inactivated vaccine viruses which are adjuvanted with alum. These, to me, are somewhat reminiscent of the Lot 100 vaccines that augmented RSV disease in infants. So, yeah, I mean, I think we're doing pretty well with the S. And I'm not sure of the great advantage of incorporating other antigens, but again, I think more research is needed.
Okay. Do we have any data yet on vaccine failures in the UK? Patients admitted to hospital more than 10 days after they've had the vaccine?
Well, I think we've been looking very hard for reinfection, and it has been documented in a number of different studies. And I think there was a lot more interest in it a few months ago, actually. And I think we all agreed that it is relatively rare to be reinfected after natural infection, and there are cases of breakthrough after vaccination, and even of very severe and possibly fatal disease. And these really need to be investigated to discover whether there is some sort of escape mutation in the virus, or whether there could be some possibility of immune-mediated enhancement. And I say immune-mediated enhancement rather than antibody-mediated enhancement, because in the experimental studies that we've done over the years on RSV, it's been quite clear that a lot of the enhancement is due to cellular immune responses in the absence of good neutralizing antibody. So it's not just antibody. It's not ADE, really. It's immune-mediated enhancement.
Okay. For one or two of vaccines that we use routinely, we do make an effort to measure the immune response. Do you see a role for testing people post-vaccine? Maybe if they've got a high level after one dose, you might not need to give them a second dose?
I think to really secure immunological memory, I would say that you do need that booster. There are vaccines which are being trialed with a single dose. So I think the Janssen adenovirus Ad26, they are saying that that's a single dose. And what they've announced is that it is protective against severe disease. Well, I think all these vaccines really protect against severe disease and death, but most of the trials, the readout is of disease reduction. That's to say any disease, because obviously designing a trial where you can have an endpoint which is any disease gets you to your endpoint faster than just looking for death or severe disease. So I think to be able to claim that you don't get severe disease, okay, but maybe a prime with an Adeno26 followed by a boost with an RNA vaccine, or maybe with Novavax purified nanoparticle. That would possibly be a way in which to sustain a really good immune response.
Okay, there's a question about the strategy of use of vaccine, which is an interesting one. Should we be using the model that was developed for Ebola and be using ring vaccination of cases as post-exposure prophylaxis as a way of preventing spread?
Well, I certainly think it's highly rational to try to vaccinate communities with very high prevalences. I was hearing about a nursing home near Norwich where they've got something like 60 cases, 60 residents, and I think only 3 of them have escaped infection. And most of the staff are infected too. And it would seem to me absolutely right to go in as soon as you have an outbreak like that and to vaccinate as many people as possible, both in the community and in anyone who's in connection with the community.
Yeah, I guess that'll become more practical when the numbers of cases are down a bit.
Yeah.
Focus on it. So anti-vaxxers. Apparently, so I'm told, that many people are worried about the long-term effects 5 to 10 years from now, and is asking, from our knowledge of previous vaccines, how likely it is that such side effects would occur. I mean, I guess this relates to the fact that we've never used an RNA vaccine before, so maybe.
Right. But we're all stuffed full of RNA. If I was to point a magic gun at you and suck out all the RNA, you wouldn't be standing well for very long. We have very efficient RNA clearance systems, both within our cells and systemically. I mean, RNA is degraded very fast, and I think there's nothing to be afraid of. There's nothing magic about RNA that is going to make us concerned long term. I mean, I don't quite get that argument that RNA is this some wonderful material that's going to cause long. I mean, I do obviously have in the back of my mind the immune augmentation stories, and with immune augmentation, you don't necessarily see it immediately. It may take some time to appear, but I think on the balance of probabilities, I personally can't wait to get the vaccines, into as many of my family as possible, ultimately, and everyone I hold dear. I would encourage them to have a vaccine. The future is uncertain, particularly with the virus that's only been around in the population for a year. But I think I see no reason to suspect that we're going to run into some terrible problem 5 or 10 years from now.
Okay. So the 70% of people would accept a vaccine, which is not fantastically high, but higher than most other European countries. How do you marry that with the fact that our infant vaccination rates are actually very, very high, well over 90%, and some of them 95%? Are we worrying about nothing? In the end of the day, will people just turn up? Or a specific question is, how are you going to convince the anti-vaxxers that they should be vaccinated?
Right. I think we could distinguish between the real anti-vaxxers whose beliefs are very hard to shift, and the large middle ground of people who, can see that these vaccines have been developed very fast and are just sort of wanting not to be at the front of the queue. They're not necessarily going to refuse to join the queue, but they just want other people to go forward first. Okay. And I think, as it becomes clear, as I hope it will and expect it will, that these vaccines are producing pretty amazing effects in terms of a downturn in disease and in transmission. I do believe that there will be a big effect on transmission as well. Yeah. And once that's evident, I think, more people at the back of the queue will start pushing forward.
Okay. We've heard a lot of data, especially recently, about the Chadox vaccine and the efficacy, and yet not so much recently about Pfizer or Moderna. I'm not sure why that is. Presumably we will be getting data. What's the latest data on why the European countries are not recommending the Oxford vaccine for the over 65s?
Right. I think, and I'm not sure so the EMA did actually recommend it, and without that restriction. And I think it was the German equivalent of JCVI that felt there wasn't enough data in the over 65s. And that is true, because the way that they did the trial in Oxford was they started with fit young people, and when it seemed safe and effective, they extended it into the older age groups. But by that stage, it was the summer, and there was very little COVID around. So I think they only had one case of COVID in the placebo and one case of COVID in the active group. So it was impossible to estimate the efficacy, but the immunogenicity in terms of the antibody generated by the Chadox vaccine was equal in the older age group. And I see no reason to therefore pull that out as a separate group and say, there is no evidence. You could pull out everyone with red hair and say, there is no evidence because there weren't enough of them in the trial. But unless you've got any reason to think that people with red hair are not going to respond, like that, they aren't developing good antibody, then, I think it's irrational actually to say that it's not going to work in the over 65s.
Okay, you did touch on this, but someone's raised the question of IgA responses at mucosal surfaces, and would that be better?
Yeah, so I'm an enthusiast for the mucosa, and I would love there to be vaccines which could be delivered mucosally as a nasal spray or nose drops, and which would educate the immune system to recognize not only that this is a pathogen, but also that it's a mucosal pathogen and that it needs a local response. So I think, quite remarkably good performance of the live attenuated flu vaccine is an illustration of how good those vaccines can be. The problem with that live attenuated flu vaccine was that it doesn't meet the conventional criteria of inducing neutralizing antibody or HA antibody in the peripheral blood. And so the regulators found it hard to accommodate within their mindset. But by measuring mucosal levels of antibody using these SAM strips, which have been developed to absorb a little bit of fluid, we were able to help them to show that actually it does induce a local antibody response, which is a good explanation for its effect.
Okay, I'm getting messages that we're going to have to wind up soon, but there's lots of questions coming through. Are any of the mutations that have been described variants, could you call them vaccine escape mutants? I guess not, in the sense they're not being driven by the vaccine.
I think that's right. I mean, they're immunological escape mutants, which may confer some vaccine resistance, but it's. I think we're still saying watch this space, but it seems very likely that there will be escape mutations which are vaccine resistant, I'm afraid.
Two patient groups of concern. One is immunosuppressed. What's the data that vaccine has any effect? And secondly, patients with severe allergies.
Right. So the immunosuppressed question, I think that the one point to make is that the Chadox vaccine is a disabled adenovirus, and it's non-replicating. So it is safe to be given to people who are immunosuppressed. It may not induce such a good immune response. The RNA vaccines should be also given to people who are immunosuppressed. But I think it's important to acknowledge that the Chadox is not regarded as a live vaccine. And there is an argument that you might need to measure an immune response in order to demonstrate efficacy.
Okay. Two or three people have asked the question about combining different types of vaccines, either in the, I guess, the same dose, or one dose of one and a second dose of another.
Yeah. So there's been animal studies just come out in the last week or so, showing that mixed vaccine approach, heterologous vaccine approach, is beneficial. And of course, when we're doing the flu vaccine campaigns, the GPs use whichever flu vaccine is available. They don't try and make sure that a person who's had a particular vaccine last year has the same vaccine this year. And actually, from the immunological point of view, presenting the antigen by different modalities may actually focus the immune response more on the antigen rather than anything else which might be present in the vaccine. And I think there's been an announcement today from Oxford that a large award has been made to some investigators there to do the mix and match approach to vaccination, so that you might, as I say, prime with say the Janssen vaccine, and then boost with the Beyondtech.
Okay, I could say, if what would your choice be? If you, I don't know if you've already had one dose, you probably have, but that would be your idea.
I'd be very happy to be in a trial, but I want to see the experiments. I'm experimentalist. I want to see the data.
Yeah. So we are going to have to wrap up, but I'm going to ask you one more question because it's been asked, and I'd also like to know the answer myself. And that is, why are these variants more transmissible? What is it about them? What's the underlying mechanism?
Yeah. Okay. So, well, that again is still being studied. I mean, there have been some studies to show that there might be enhanced binding to the receptor, and of course, there's not only one receptor, but a second receptor that's been described now. So, need to look at those bindings, and then replication. So there are reports that the new variant, the B.1, 0.7, does actually replicate better in some tissue culture environments. So it could be that each droplet that comes out of the nose has actually got a higher concentration of virus. I think those are the most likely. I mean, it could also be that it could cause more particles to be expelled, I guess, but there wouldn't be any particular selective advantage in adapting in order to generate greater severity of disease, as far as anyone can think. Yeah. Okay.
I've not got through these questions completely, but I am going to have to wind it up now. So thank you very much indeed for the talk and for my interrogation. You responded extremely well. I wonder if some of these questions that we haven't asked, if we pass them on to you, if you'd be willing to have a look at them, and maybe
I'll see if I can do that. Yes, thank you.
Yeah, we should have asked you that before we started. So it just remains for me to thank everybody. We were around about 150 people, which is great. Fantastic talk. Next week is long COVID, so do re-register for that if you're interested in that. Lots to know about long COVID that we don't know yet. But that's it. Thank you very much again, Peter, and good night to everybody. Hope to see you next week.
Thank you very much.